So some molecules need to be moved against their concentration gradient. This is where we'll consider active transport. Active transport is active because it requires energy, usually in the form of ATP, which can either be used directly or indirectly to fuel active transport. We'll look at both examples. In active transport, we are moving substances from low concentration to high concentration. That is against their concentration gradient. So we're no longer going with the flow passively downhill. We have to use energy to move molecules uphill against their gradient. This requires the use of very highly selective carrier proteins. The carrier proteins that we use in active transport come in three different types. First of all, uniporters. They move one molecule at a time in one direction. Next, we'll see symporters. Symporters move two molecules at a time, but in the same direction, as you can see in the middle here. And then there are antiporters that move two molecules in opposite directions across the phospholipid bilayer membrane. The prime example of active transport is seen in the sodium-potassium pump. We use ATP directly for active transport in this mechanism. It uses an antiporter to move 3 sodium out of the cell and 2 potassium into the cell. That's the key you'll need to remember. 3 sodium out and 2 potassium in, both of which are moving against their concentration gradient. The whole process begins with the membrane carrier protein opening to the inside of the membrane and binding 3 intracellular sodium ions. ATP phosphorylates the protein, that is, it gives its energy to the protein, which causes a conformational change, pushing out the 3 sodium ions against their gradient into the extracellular fluid. Now, potassium can bind. 2 potassium bind to the protein whose conformation is open to the outside of the cell. And then, once these have bound, the protein once again changes conformation and dumps the potassiums into the intracellular environment. Both sodium and potassium are being moved against their concentration gradient through the use of ATP as energy. Because of the fueling of ATP and the change in conformational change, the affinity of the carrier protein for either sodium or potassium changes so that the ions can be carried across the membrane against their concentration gradients. When it's open to the inside, sodium binds most easily, and when it's open to the outside, potassium binds most easily. So 3 sodium out and 2 potassium in. Now let's look at coupled transport or secondary active transport. Secondary active transport or coupled transport uses ATP indirectly. So let's consider the gradient that's generated by the sodium-potassium pump. We just looked at this one. ATP is used to fuel the movement of 3 sodium out and 2 potassium in. This creates a gradient of sodium. We're pushing sodium against its gradient outside, so there's lots and lots of sodium outside. Now that sodium really wants to move back inside the cell any way it can. And as it happens, there's another transport protein. However, this transport protein is for the transport of sodium and glucose together. So sodium is going down its concentration gradient. And because this carrier protein is specific to both sodium and glucose, glucose needs to go with it. So sodium and glucose are both transported together back into the cell based on the concentration gradient of sodium that was created by the sodium-potassium pump. This is why energy replacement drinks, like Powerade or Gatorade, come with sodium as well as glucose, because sodium and glucose are transported together across the gut. To put a little sodium in the drink, a little electrolyte in the drink, allows much more efficient transport of glucose into your cells. This coupled transport uses the energy that's released because of the transport of sodium. So sodium is the molecule that's moving by diffusion after it's been powered by ATP over here, in order to actively transport glucose into the cell, because glucose is moving against its concentration gradient, but only because of the gradient we created with sodium. Lots of sodium outside, lots of glucose inside, but this transporter is open for glucose and sodium together, so we get to move glucose against its gradient. In this case, the ATP is used indirectly, thus we call it secondary active transport. The sodium glucose transporter is a symporter, which captures the energy of sodium rushing back into the cell to move glucose against its concentration gradient. Let's take a moment to view another animation to clarify active transport.
When substances need to be driven against their concentration gradient from low to high concentrations, active transport is required. This process uses energy to move a substance across the membrane using selective protein carriers. The sodium-potassium pump is a specialized membrane protein carrier, common in most animal cells, that moves sodium and potassium ions across the plasma membrane. ATP fuels the pump in the movement of these ions from low to high concentrations, moving sodium ions outside of the cell where they become concentrated and bringing potassium ions into the cell where their concentration is higher relative to the outside. In other instances of active transport, the concentration gradient created from the action of one pump can be used as the input of energy to drive a different pump, a process known as coupled transport. The large concentration gradient of sodium ions is used to bring glucose into the cell against its concentration gradient.
Our final means of transport is bulk transport or bulk passage, and it's composed of endocytosis or exocytosis. Endo meaning inside, cytosis the cell, so to bring inside the cell, and exo to take outside of the cell. So endocytosis involves movement of substances into the cell, and it happens in one of two ways, either phagocytosis or penocytosis. The root phage means to eat. So the literal meaning of phagocytosis is cellular eating. Penocytosis is cellular drinking, and so it takes in only fluid and the solutes that are in the fluid. Now, we may see receptor-mediated endocytosis. This is where specific molecules are taken in after they bind to a particular receptor. So in this case, we have a target molecule and receptors for these target molecules. Once the target molecule binds to the receptor, a vesicle forms and engulfs those target molecules. The human genetic disease of familial hypercholesterolemia, the LDL receptors lack any tails, so they never fasten to the clathrin-coated pits. So that is, these Y-shaped guys never actually fasten into the pits. As a result, it doesn't trigger vesicle formation, and the cholesterol is not taken into the cell, but rather it's left out in the bloodstream. And here it accumulates as plaques inside the arteries and leads to heart attacks. So LDLs are our bad cholesterols. And if we have lots of extra LDLs or bad cholesterols floating around in our blood, then we're much more susceptible to heart disease. Exocytosis is the movement of materials out of the cell. It's used in plants to export the materials they use to build their cell walls. And in animals, we use it to secrete hormones or neurotransmitters or digestive enzymes, or maybe even waste products that can be picked up by the blood and then excreted from the body. Okay, time for the final card quiz of this chapter. A macrophage engulfing a bacteria is an example of what type of transport protein can move two different molecules in the same direction? How is the sodium-potassium pump able to move ions against their concentration gradients? Here you go, green, blue, and red. Those ones were pretty straightforward, but if you missed one, please go back and investigate. See if you can fill in the gaps.